We're fortunate to have Professor Wilfred van der Donk from the University of Illinois, where he is the Richard Hecker Chair in Chemistry. A little bit about Wilfred, who really has done some spectacular work, particularly in the lanthionine peptide area. He received his both BS, MS at the University of Leiden. His PhD was in the States at Rice University, and he did a postdoc at MIT with Joan Stubbe. And he has been at the University of Illinois since 1997, where he's also been a Howard Hughes Scholar since 2008. And the title of his presentation is Combinatorial Methods Towards Genetically Encoded Cyclic Peptides.
Thanks so much, John, for the kind introduction and also for the invitation. This is actually my first time that I'm at the Boulder Peptide Symposium, and I've really, really enjoyed it. I think the format that you guys have is excellent, and I've learned a lot about questions that I typically don't think about, especially yesterday's workshop was eye-opening for an academic. That also made me decide that I should put in a disclaimer here, in that you're going to hear typical academic research in the next half hour or so. And so we try to understand new systems, we try to develop new tools, but I have to admit that we don't think yet about some of the questions that I've heard about this week. And certainly at some point we or people that will translate our methodology will need to do so. So we've heard a lot this week about cyclic peptides. And we heard in Dan Rich's talk in particular also about natural products, cyclic peptides. And so in the mid-90s, when the biosynthesis of cyclic peptides started to become clear through non-ribosomal peptide synthetases, there was a lot of excitement that perhaps we could borrow those methods from nature to make analogs or to make even non-natural molecules. I think it's fair to say that that has been difficult. These non-ribosomal peptide synthetases are very large. They are these mega syntheses, assembly line enzymes, and engineering them has turned out to be difficult. Now, over the past decade, as many bacterial genomes and also fungal genomes started to come out, it became clear that a different way by which nature makes cyclic peptides that was believed to be a very small niche area turns out to be actually very large, and nature ubiquitously uses the ribosomal route to cyclic peptides. Many compounds that we thought were non-ribosomal, we now realize are actually made ribosomally. So first in just a cartoon format, a little bit about the strategy that nature uses. So nature first makes a linear peptide that is DNA encoded, and then carries out a series of post-translation modifications, introducing stable chemical structures that are not found in the 20 natural amino acids on part of that peptide, which is called the core peptide. Now there's typically an N-terminal extension and sometimes a C-terminal extension, and these are removed in the final step of biosynthesis, and then we have, which the community has decided to call now a RIP, for ribosomally synthesized and post-translationally modified peptide. And so you can immediately see that there's a lot of advantages to a system like this that nature certainly has recognized. These are very, very evolvable. I think much more evolvable than any other natural product, because all you need to do is make a change in the DNA sequence to get a change in the core peptide, and you have a chance for a new natural product. Now, that would only happen if the enzymes would tolerate changes in the peptide. And of course, most enzymes, perhaps not the one we just heard about, but most natural enzymes tend to be very specific to their natural substrate. And so nature has done something very interesting here. It has separated substrate recognition from chemistry. And so the substrate recognition is done by the leader peptide and or the recognition sequences. And as a result, the active site of the enzyme can be very forgiving with respect to the sequence of the core peptide. And I'll show you some examples of hypervariable core peptides in nature. And so what nature can do then, it doesn't have to worry about the specificity of the enzyme. The enzyme is not going to go crazy on all kinds of peptides in the cell, because only when the leader peptide is attached will the enzyme act on it. And so many of the enzymes that we and others have studied are very, very tolerant with respect to the sequence of the core peptide. Now, of course, these attractive aspects are immediately clear, are also very attractive if you want to make cyclic peptides or you want to change the structure of the cyclic peptides. Also attractive for genome mining, the current direction of natural product synthesis and biosynthesis, where you no longer find the molecule first, but you find the genes first, and then you express those genes in a heterologous host in order to get to the natural product. And then I'll show you some examples of synthetic biology, although there's a lot more things that we and others have done that I won't have time to tell you about. I decided to try and focus on things that I thought you might be interested in. So some chemical structure. So there's now more than 30 different natural product classes, each of which has thousands of structurally diverse members that are made by this type of overall biosynthetic strategy. We work on the thiopeptides. I won't have time to tell you about that today. Here is nature's version of stapled peptides. These are alpha helices held together by covalent links between cysteines and the alpha carbons of amino acids elsewhere in the peptide. Eric Schmidt in particular has been working on the cyanobactins. These are N-to-C cyclized peptides that sometimes but not always are decorated by these heterocycles. David Craig has really done terrific work on the cyclotides. David is here, I see him there. And so he has shown that these systems are very, very amenable to grafting of pharmaceutically important sequences. And then you heard earlier this week already about the lysopeptides, also a class of very stable peptides that can also be used for grafting. So in my talk today, I will focus on the lanthipeptides, as John mentioned. This is the system, the RIP system that we've been working on the longest. We're working on about 5 or 6 different RIP classes now. And so niacin is the best known example, and many of you may have known about niacin because it has a long history in peptide chemistry. And it contains 5 rings, 5 thioether rings, and the one in red here, a lanthionine, has given the lanthipeptides their name. Niacin is used in the food industry ubiquitously in order to combat foodborne pathogens like Clostridium and Listeria. And so, as you heard in the introduction, I did my postdoc with Joanne Stubby. That's where I became interested in enzymes. My PhD training is actually in synthetic chemistry. And so we wanted to understand the enzymes that make niacin, because I was really intrigued by the fact that you need only 2 enzymes to make this rather complex natural product. And so these 2 enzymes do 2 simple reactions: elimination of water from serines and threonines that then generates these electrophilic species. And then a second enzyme adds the cysteine side chain to the beta carbons of the dihydroalanine and the dihydrobutyrene that first generates then an enolate, and the enzyme stereoselectively protonates that enolate such that we have the D stereocenters at all of these junctions here. And so I'll use this shorthand notation in some of my slides. So in a little bit more detail, this is the precursor peptide that comes off the ribosome. We call that Nis-A. And then a single enzyme comes in and it dehydrates all red and blue serines and threonines. Then a second enzyme comes in and it catalyzes the stereo, chemo, and regioselective cyclization process, such that, that after the cyclization, a protease can come in, can remove the leader peptide, and we have niacin. And so we were really intrigued, again, as enzymologists, as to how 2 enzymes could do this, how the enzymes could be so specific with these serines and threonines, but not touch the serines and the threonines in the leader peptide, how one enzyme can do all of these cyclizations to just give you one product, whereas non-enzymatically we tried and we get a total mess. Now today I won't focus on the data that we have obtained in order to understand these enzymes. I wouldn't say that we completely understand what's going on, but we understand a lot about the dehydratase and the cyclase. Instead, I want to focus on a rather unusual system that we discovered about 5 years ago, maybe 7 years ago. And for that I want to introduce the class 2 lanthipeptides. So rather than having 2 enzymes that carry out the dehydration and the cyclization, the 2 reactions are carried out by a bifunctional enzyme. That's helpful because now we only need to worry about a single biosynthetic enzyme. And that enzyme has a dehydratase domain that uses ATP to phosphorylate the side chains of the serines and the threonines. And then it has a cyclase domain that is used in order to do the Michael type addition. And so from everything that I'm going to talk about from here on are systems like this, where we have a single enzyme that introduces all of the rings. So Bo Lee, a student at the time in my lab, did some genome sequence analysis. As I said, we are very interested in genome mining. And in 2009, she found a system in a genome that had just come out from Prochlorococcus. And so this is a marine cyanobacterium. It's the third most abundant organism on Earth. It was discovered by Penny Chisholm at MIT, and she also reported the first genome sequences. And we saw a system that looked really intriguing to us, because it seemed like encoded on the genome was just a single class 2 lanthipeptide synthetase. But as you can see, there's 30 little genes that encode for peptides that to us looked like any of them could be the substrate for the enzyme. We decided then to start a collaboration with Penny's lab. She sent us genomic DNA. Bo cloned out the enzyme and all of the substrates. And to show you why we were kind of intrigued by it, the leader peptides are almost identical. Not quite identical, but very, very high sequence similarity. And that's why we thought any of these could be the substrate. But when we then look at the core peptides, where the enzyme is supposed to do its chemistry, you can see there's very, very high diversity. In fact, there's not a single pair of peptides that has any homology with one another. And so we were intrigued and wondered whether one enzyme could really act on all of these peptides, and that turns out to be true. So the single enzyme will take these substrates and turn every single one of them into a polycyclic product. So the organism that produces this, we can get maybe 20 micrograms out of 10 liters of a mixture of peptides. So we can see that they're there, but we could never determine their structure. And that's really why we developed methodology that now turns out to be very, very useful for us, which is to make these molecules in E.coli. And the system is really very simple. You simply co-express the his-tag precursor peptide with all of the biosynthetic enzymes, in this case just a single one, but we've done this for now 70 different lanthipeptides, and we can go up to 5 or 6 different biosynthetic enzymes that further decorate peptides, like with chlorination of hydroxylation or something like that. Now we don't put in the protease. That way the leader peptide stays on after the post-translational modifications. And that gives us the advantage that we're not making an antibiotic. We're not killing the E. coli. So this is just fortuitous. Removing the leader peptide is actually arming the molecule. That gives us then the additional advantage that we can put a his tag on that leader peptide, and we can do a one-step affinity chromatography purification. And those of you that have perhaps purified natural products know what a great advantage it is that you can have a one-step purification compared to having to do a lot of chromatography. Now we then do have to remove the leader peptide in order to get the natural product. And it turns out that, again, nature is very forgiving for us. And typically we can put a commercial protease cleavage site between the leader peptide and the core peptide and just buy a protease. And while the peptide is still bound to the nickel column, we cleave off the leader peptide and our product comes off the column. And so, as I mentioned, we've done this now for more than 70 different lanthipeptides, showing just a few here. So I'll tell you more about the prochlorosin. So this is one of those 30 different peptides. But we can also do it for Actagardin, which has now passed phase 2 clinical trials against Clostridium difficile. We can do it for duramycin, which has also successfully completed phase 2 clinical trials for cystic fibrosis. And we can also do it for that commercially used food preservative. So it seems to be very general and seems to work quite well. So going back then to the 30 different peptides, my student Wei-Xing Tang chose 6 of them, made maybe 20 milligrams of it in E. coli so that she could determine the NMR structures of all of them. As you can see, these NMR structures tell us that the topology of all of these products are very, very different. Such that we can have rings that are non-overlapping, we can have rings that are overlapping, these tricycles, we can have very small rings, we can have very large rings. So again, as an enzymologist, this looks kind of strange. I mean, how can one enzyme do all of this? Because usually an enzyme will catalyze certain reactions over competing reactions, But it's hard to imagine how you could engineer or even evolve an enzyme that could do that for 30 different substrates. So we first decided to investigate how the enzyme might do that. And then later in the talk, I will also tell you about what we have done with an enzyme like this that is intrinsically promiscuous and can make a lot of different products. And so I want to, so these are the two things that I'm going to finish with. And I want to highlight this prochlorosin 2.8, since that's one that will be coming back a number of times. And that is because for no really good reasons, my students did much of the work on prochlorosin 2.8. It's one of the first that we made in E.coli where we determined the structure of. But what I'm about to show you, we've also shown for other peptides, but it's not as far advanced as it is for prochlorosin 2.8. Okay. So as Dan said on Tuesday, I can't help myself. I'm an academic, and so I want to tell you a little bit about how this might work. And so at first we thought that maybe this enzyme wasn't a very good enzyme and it would be making mixtures. But as you can see, it's not making mixtures. For each peptide, it makes a single product. Maybe a little bit of other ring topologies that are in the baseline. But by and large, despite the fact that these ring topologies are all very different, the enzyme is really good, at least in E. coli. And we suspect that the same is true in the producing organism. So we decided to investigate a little bit as to how this might happen. So two of my students, Shubha Mukherjee, now at BMS and EU, investigated a number of different possibilities as to how one enzyme might do that. So this enzyme has only 2 active sites, so there's not like a number of different active sites that are there for each and every single substrate. And so first we thought that maybe the cyclizations would be non-enzymatic. It's not that difficult to add a cysteine to a dihydroamino acid. And so I won't go into the details, but Shubho did a lot of solid phase peptide synthesis to make protected peptides, to then give those to the enzyme, have the enzyme introduced to dihydroamino acids. Then our protecting groups are photolabile, so we can shine light on it and ask, is there a place for non-enzymatic cyclization? And the answer is no. The non-enzymatic cyclizations will take place, but they're A, way too slow to be involved in the enzymatic reaction, and B, they give us a mixture of compounds. So that's not what's going on. We then thought that perhaps the enzyme simply equilibrates the product, so where there's a Michael addition and a retro-Michael addition, and it keeps doing that until the lowest energy products are formed. And so again, to test that, we did a lot of solid phase peptide synthesis. Shubha would make intermediates and products that would have the wrong topology, then feed those to the enzyme and ask, can the enzyme correct those? And there the answer is also no. So we don't believe that that is what is going on. So we currently think that the topology that is formed is actually not determined by the enzyme, but rather by the substrate. And so we have some pieces of evidence for that. I wouldn't say that we have proven this, but I will show you one of those suggestive pieces of information in the PNAS paper. I don't have time to tell you about the Nature Chemistry paper. So what we decided to do is that if the information is in the core peptide, in principle, it shouldn't matter what enzyme is then present. And so we decided to try the following. Take the leader peptide of that very promiscuous system and then fuse to it at the DNA level the core peptide of a completely different lanthipeptide. And so ProcM and its leader peptide come out of the oceans, as I mentioned. Lactosin 481 is an antibiotic that is made in the udder of the cow. And so we were wondering, if we have this chimeric substrate, will the ProcM in E. coli take that core peptide and make lactosin 481? And so here is physically showing what we're trying to do. So here are the 6 peptides of which we know the structure. There's, of course, 24 other substrates, but none of them look like the core peptide for lactosin 481. And its cognate enzyme turns it into this cyclic peptide. And so here's the result of the experiment. Here is authentic lactosin 481 that was isolated from the Lactococcus lactis. So we put it down in the middle here. These are agar growth diffusion assays. So where it's opaque, we have an indicator strain growing. We put the compounds down in the middle, and where you see the growth, the zone of growth inhibition is where the compound diffuses out and kills the bacteria. As you can see, authentic lactosin 481 and lactosin 481 made by our technology in E.coli are about the same. The product from this experiment seems to have a much larger zone of growth inhibition, but that's because my student didn't anticipate much. So she thought if I even get 2 or 3% of the right product, I want to catch it. So she put down more material. We now know that about 75 to 80% of the product that comes out of this experiment is lactosin 481. And that is remarkable because if you just do the calculation, you can make more than 50 different products with the number of dehydroamino acids and cysteines that we have in this peptide. Yet this enzyme that has never seen this makes the correct product in about 75 to 80% yield. So to us, that gives very strong suggestion that the enzyme doesn't really know what it needs to make, but that the information is actually in the peptide to make the topology that we see in the end. So what can we do then with an enzyme like this? And again, I'm going to show you almost everything for prochlorosin 2.8, but we've done similar studies with other prochlorosins, but they are not as far advanced. And so what Xiaoyang decided to ask is, can we make libraries of cyclic peptides? From here on, everything is unpublished. This is working out. We change residues between the and the serines, as you can see here. So here is the natural sequence of 2.8. And we would like to know if we randomize these sequences, will the enzyme still be really good? Now, we did have certain number of restrictions. We did not want to introduce more serines, threonines, or cysteines because we were worried that that could mess up the enzyme. And if we are right, if the folding landscape of these peptides on the enzymes is what leads to single products, then we also didn't want proline or glycine. And so we looked at the codon usage for codon usage that wouldn't introduce any of those 5 amino acids, but as many other amino acids as possible. And so at the DNA level, if you make a library and you use the NWY codon, where N stands for any of the 4 nucleotides, W is A or T, and Y is T or C, then you can go to the genetic code and find that you can encode 8 different amino acids. And for each amino acid, there will be 2 codons. So you get nice distribution of those 8 amino acids. Now, there's no positive amino acids in here, no positively charged amino acids. So you can also go to VWK. In that case, you can also introduce lysine. You do introduce some bias for these 2 hydrophobic amino acids over other amino acids. What I'm about to show you was all done with the NWY codon usage for reasons that will become clearer later on. So at the DNA level, we then order the oligonucleotides, put them into a plasmid that is a co-expression plasmid of the enzyme, and each plasmid then has a different sequence of our substrate. We transform E.coli with them, and then isolate the plasmid and sequence. And as you can see, we don't get quite as even distribution as theoretically you would expect. There is some bias. But we're pretty pleased by what we get out. So these are the different positions that we have randomized here. And you can see the distributions of the different amino acids. Now, I want to say that we have here a little bit more than 8 million unique peptides. But the theoretical diversity of having 10 positions and having 8 different amino acids is significantly higher. So this will be 0.8% of the theoretical diversity, and that will become important later on. So we first wanted to make sure then that the enzyme would actually be good, that we wouldn't get only a very, very small number of correctly cyclized products. And so we picked a number of colonies and analyzed them by a number of methods. So the MALDI mass spec immediately tells us that there's 2 dehydrations, so that's good, because we need 2 dehydrations. Here's the original sequence again. And then to test whether indeed we have formed the 2 rings, we test by reacting with NEM. If there is a free cysteine, we'll get an adduct. If a thioether has been formed, we will not get an adduct. And as you can see, after we add NEM, we don't see any adduct formation. So this is a cyclized peptide. And then we confirm that with MS and show that indeed the topology is correct. So we did this for almost 40 different clones that we picked. You can see the sequences here are indeed highly diverse. and all of these peptides are fully dehydrated and are fully cyclized. And when we picked another 25, that is still the case. So to date, we have not found an example of that library that is not dehydrated and cyclized. But of course, we've not picked everything, so I can tell you with a 95% confidence that 85% of our library is dehydrated and cyclized. So we think that that means that we have a high-quality library. So then what can we do with that? So we can clearly make good libraries. And so we decided initially to not only want to get binding to something that we're interested in, but also a functional readout. And so for that, we decided to go to a reverse 2-hybrid system that was developed in Steve Benkovic's laboratory by Ali Tafazoli, who now has his own laboratory. And so we collaborate with Ali. And so the way it works is that you have a protein-protein interaction of interest that is attached to a repressor. And so as long as the protein-protein interaction is there, we are not making an essential histidine biosynthetic enzyme, and we're not making a kanamycin-resistant enzyme. So if you plate these E. coli on agar that contains kanamycin and that lacks histidine, you get a severe growth defect. At some point you will see some growth because you get some mutations here, and that will solve the problem of not being able to make histidine and being killed by kanamycin. But by and large, it takes many days before you see growth here. Now, if in each cell you express a different cyclic peptide, and if one of these peptides disrupts that protein-protein interaction, you get much accelerated growth. So all we do is we take our transformed library, plate it onto a plate that has kanamycin and lacks histidine, and we just see what grows up. And then we pick those colonies. We then have 2 counter selections because we don't want non-specific interactions. And we also don't want cases where we did get mutations, for instance, there, or some other mutations that are not related. But it works really, really well. I mean, if you are willing to take a certain number of positive, false positives, which we definitely get, the system works quite well. And so the one I will tell you about is a protein-protein interaction between a human escort protein, or its domain called UEV, and the HIV polyprotein GAG and its interaction with the P6 peptide. So this part of P6 interacts with UEV, and it's a very well characterized interaction. It's an interaction that has a large surface. We thought it would be a good way to see whether our methodology will actually work. Now, we made one big mistake, though. We chose an intracellular target, and that meant that later on we were asked by reviewers, okay, is this actually active intracellularly? So in retrospect, we should have, for our first examples, chosen extracellular targets, but we didn't. So Ali has about 12 of these reverse 2-hybrid systems developed, and so we're now doing the similar type of selections, but now for extracellular targets as well. So the native interaction, if you synthesize this nona peptide, it binds to the UEV domain with KDs that vary between 3 and 50 micromolar, depending on how you do the assay. In our hands, it's 16 micromolar. And if we then take the best hit that we get, in this case from 2 million cyclic peptides, it's about 10-fold better than this green nona peptide. So we're quite happy about that. I know that many of you look for nanomolar or even picomolar interactions. But this is our first hit out of 0.2% of the theoretical diversity of a library like this. So I'll tell you in a few slides how we're now trying to do affinity maturation of this hit. Let me first show you what the hit looks like. So here is the structure. And so as expected, it has 2 rings. The sequence is shown there. It binds, as we show by biophysical methods, to the human protein, not to the HIV protein. And we need cyclization. If we take the linear peptide, we see no binding. It's also sequence specific, because if we invert the sequence of either the B ring or the A ring, we also do not see any binding. Now, what about then activity in cells? And so our peptide is not cell permeable, but we were still able to test the activity in cells by using a trick that I'm sure you're all familiar with. In our case, we attached the TAT cell-penetrating peptide to an additional cysteine that we incorporate. We were worried that that cysteine might do something, but it turns out that it doesn't. And so in the cell, this disulfide is reduced, and then we can use a standard HIV budding assay, which uses GAG that is GFP labeled, and you see how much of the GAG is forming virus-like particles. And the TAT peptide itself doesn't really do anything. At about tenfold under the KD, we see significant buildup of the GAG GFP inside the cells. And we start seeing a reduction of virus-like particles that are coming out of these HEC293 cells. Now, this is, as I said, well under the KD, and so when we go to higher concentrations, we can completely shut this down, and everything stays in the cells. But unfortunately, we start running into cytotoxicity. So at 1 micromolar, we can prevent the formation of virus-like particles, but we clearly start having cytotoxicity. We've investigated why that might be, and it turns out that this is, if you wish, on-target cytotoxicity, because the UEV domain of these escort proteins is not there to interact with HIV. HIV is hijacking this. What the UEV domain normally does is bind to a very similar motif on the HRS protein in humans, and that is important for effector downregulation. So the EGF receptor, after signaling has taken place, it needs to be removed, and it's this escort protein that is involved in the ubiquitinylation and putting it into endosomes. And so we can show that our peptide indeed interferes with the receptor downregulation, and that is a toxic activity. So we don't currently have any therapeutic window, or very small therapeutic window. So coming back that we've only really interrogated a very small percentage of the theoretical diversity, can we improve this then? Can we get perhaps more tight binding? And can we also distinguish these two processes here? Now, it turns out that our peptide is non-competitive, so it doesn't bind at the same place as the PTAP peptide. Again, this is kind of nice from these functional two hybrid assays. You know that it disrupts the protein-protein interaction, but whether it's competitive or allosteric, we don't know. And it turns out that in our case, it's probably allosteric. We don't have a structure yet of the peptide bound to the UEV domain. But if we want to do maturation, we cannot use these techniques. We cannot, keep doing the selection over and over. So ideally, now that we have a hit that binds, and it binds at a place where it disrupts protein-protein interaction, we would like to use display techniques to improve it. And so we have developed this. I will show it just for yeast display, but we also have done both phage display and mRNA display. And so for yeast display, Mark Walker essentially did the following. He puts the precursor peptide attached to the AGA2 protein. So this is well-known technology that was actually developed at the University of Illinois when Wittrup was still there. So if you express this inside the yeast, and you also express that one enzyme that does the dehydration and the cyclization, the question is, will the enzyme be good enough to get all of the post-translational modifications done before the export system puts this on the outside? So this is something that you usually don't have to worry about with display systems, but we of course do. So we chose what we consider a difficult system. So for haloduricin, this is an antibiotic. We need to do 7 dehydrations and 4 cyclizations. But it turns out that if we then look what happens and we cut from the yeast, the final product, that it is fully dehydrated. It has biological activity, antimicrobial activity, the same as the authentic material, suggesting that we get full cyclization. And again, we can do a test, in this case an iodoacetamide test, showing that the displayed peptide is indeed fully dehydrated and cyclized. So we're using this now to take our first hit that binds to UEV, then use fluorescently labeled UEV and use FACS analysis in order to see if we can improve the binding, and we can do counter selections then with the HRS protein. And as I said, we've also done this for phage display, and with Hirozuga at the University of Tokyo and Yuki Goto, we've also done it for mRNA display. So these enzymes are very, very forgiving in the context that you want to use them. So let me summarize then. So I think in general these RIPs are really, really intriguing and perhaps very, very interesting for translational applications. We have shown it for the lanthipeptides and a few other RIP classes. I also want to mention Eric Schmidt's work, who has done it for cyanobactins and other cyclic peptides, monocyclic peptides. And he has also shown this very high tolerance to the substrate. These pathways are very portable, so you can do it in E. coli, you can do it in yeast, we can do it in streptomyces. And again, that's because everything is DNA encoded. We only need DNA, polymerase, a ribosome, and the 20 natural amino acids. So we don't need any building blocks here. And I didn't have time, I threw it out yesterday, but we can also do rational design. So we can bring in enzymes from 2 different RIP classes and make them act on a single peptide by knowing something about where the leader peptide recognition sequences are, and make chimeric leader peptides, and then make, for instance, hybrids between thiazole-containing peptides and lanthipeptides. And so I need to thank a really terrific group of co-workers in my lab. That has been really a pleasure to work with. Also want to thank the funding agencies, all of the collaborators that have been really terrific. And finally, thank you for your attention.